Now we're going to cover the phylum Cnidaria. There are some other animal phyla that are related to the Cnidaria because they have two tissue layers as well. But we're going to just focus on the Cnidaria as far as, making drawings and learning details and all that. So the Cnidaria, before we can really understand the Cnidaria, we need to understand about how animals develop. Because the sponge had no tissues, and the Cnidaria have two tissue layers. They're what we call diploblastic, or they're called a diploblast. And instead of tissue layers, we're going to say that they have two germ layers. So this is where we need to talk about development, and this is with any animal except for animals as simple as a sponge, because the sponge don't have tissues. But when you get into animals that have two tissue layers or more, their development starts out with a fertilized egg. We call that a zygote. This is the egg, this is the sperm. That sperm has DNA from the mother and the father. I'm so sorry. I'm not going to redo that, but I was thinking about sperm fertilizing eggs, but this is not a sperm. This is a nucleus. Okay, so this is I better start labeling things. This is our zygote, which is a fertilized egg. And the nucleus of this zygote contains DNA from the sperm and contains DNA from the egg. So the zygote is the first cell that's going to form into the organism. The zygote is going to undergo mitosis. And it's going to form a whole bunch of cells just like the zygote. They're going to be clones, and we call these cells embryonic stem cells. Okay? They're eventually going to form into a ball of cells that is hollow on the inside. And I'm just going to draw it like this, and then draw these dividers for my cells. I'm drawing this like we're looking into the ball of cells, or the blastula. This blastula is also composed of embryonic stem cells, but now they have formed a hollow ball of cells called the blastula. And then that hollow ball of cells, I'm just drawing the nucleus in here, so they look like cells. That hollow ball of cells is going to start to make an indention. And it's going to make an indention, and that indention is going to become the digestive tract. So I'm not going to go so slowly that I go through every single step for you, but hopefully you can understand that these are embryonic stem cells. They're all alike. But if an animal is going to have arms and legs and a brain and a liver, then some of the cells are going to have to differentiate or specialize. And they're going to have to form different types of tissues, which will then form organs and organ systems. So it has to be more complex than just embryonic stem cells, which are all identical to each other. So what's going to happen is this ball of cells is going to form an indention, and that's going to, once it kind of dents in or forms an indention, curves inward, then we're going to call that a gastrula. But I'm just going to go straight into the tissue layer part. So for a diploblast, there are cells that are going to form on the outside, or there's tissue that's going to form on the outside called the ectoderm. And this is before any major structures form. You can see that. Sorry. The lighting's not real good. Try to get it where you can see. And my drinks are in the way. Okay. All right. Hopefully you can see that. So that outside layer, I'm just going to color that layer blue. That's going to be our ectoderm, and it's composed of cells. And that ectoderm is going to form into the outside layer of the cnidaria. And the cnidaria can be a jellyfish, or can be a coral, or a sea anemone, or even an animal that lives in fresh water called a hydra. There are different types, but we probably relate more to the jellyfish than anything. So that's going to be the outside layer, and then the layer that lines the digestive tract, our inner layer, is going to be the endoderm, and it's going to be in here, and there's going to be a hole in the middle, and that hole represents all the way from the mouth to the anus. Or in the case of the cnidaria, there's only one opening that forms. There aren't two openings. So this is called an incomplete digestive tract. There's only one opening. That one opening is the mouth and the anus. So this is the endoderm that's going to line the digestive cavity. And then you're going to have some jelly-like stuff in here called the mesoglia. And this is what makes it, squishy like a jellyfish. The mesoglia doesn't have cells. It's not cellular. It's just a chemical that's jelly-like, because the substance that's jelly-like. Now, what's going to happen is eventually this diploblastic organism is going to start developing tentacles, and it's going to start looking more like, its cells are going to specialize, and it's going to start looking more like either a jellyfish or a sea anemone. So what we have with that is we have two body forms in the cnidaria. The jellyfish has the medusa body form, and I did the ectoderm in blue, so I'm going to keep that up. So this is going to be the medusa body form, and this is the ectoderm. And then the ectoderm is going to kind of come around like this. And then here's an opening. Okay, so that's where the ectoderm is. This is the both mouth and anus. And then it's going to be lined by, on the inside, it's going to be lined by endoderm. It's going to kind of sort of follow the same pattern. Oh, sorry, I didn't mean to do that. But anyway, it's going to be lined by endoderm. The purple is the endoderm. So our blue is our ectoderm, which is like the skin. And our purple is the endoderm, which lines the GI tract, lines the digestive tract. Now, what we don't have is a mesoderm. We don't have a middle layer. All we have is this jelly. In fact, I'm going to draw it more circular so it looks more like jelly. Just draw these little squigglies. All in here, this is mesoglia. So it's just a tissue that's jelly-like, but it's not, I shouldn't use the word tissue. It's just a substance that is jelly-like, but it does not contain any cells. All right, so that is the medusa body form that we find with the jellyfish. And what we see here are the tentacles are facing downward. Okay? The tentacles and the mouth/anus are facing downward. And then we can have a what's called a polyp form. And this is like a sea anemone. A sea anemone would have the polyp form. Okay, so let's draw that kind of like this, sort of box-like at the bottom. And I'm just going to draw two tentacles, but know that they come all the way around. I mean, there's tentacles all the way around, but I'm just drawing two because I want you to be able to see the, I don't know why they're different sizes. They should be the same size. But I want you to be able to see the lining of the digestive tract. So the blue is the endoderm, The purple is, I said that wrong. The blue is the ectoderm, the purple is the endoderm. And notice that it's going to come kind of go down like this. And then this hole is the mouth/anus. This is the ectoderm, the blue, and then the purple is the endoderm. And same thing as before, the mesoderm, I'm sorry, the mesoglia. I didn't label that up here. Let me go ahead and do that. It's not mesoderm, it's mesoglia. It's the jelly-like material that fills the gap in between the endo and the ectoderm. So this is the polyp form, the polyp body form. The tentacles are facing up. Okay. All right, and I'm good with you just making these two drawings. There are some specific cell types, but I'm just going to have you answer the questions about those. The way that the all of the Cnidaria feed, they are predators. I'll just tell you this information. There isn't really any more drawings. The Cnidaria are predators, but they really aren't. They're not just going around looking for food. They just have sensory receptors. So when an animal touches up against them and they feel it, that triggers their stinging cells called cnidocytes to release nematocyst. And nematocyst is the stinger that contains a toxin. And that will paralyze the prey, and they will take the prey into their mouth/anus. So they are definitely predators. They are definitely carnivores, but they are only diploblastic. They only have the two germ layers.